Welcome to another episode from the Veterinary Research Channel. In this episode, we delve into a fascinating area of veterinary medicine, fecal microbiota transplantation, often referred to as FMT. This procedure, which involves transferring fecal material from a healthy donor to a recipient, is gaining increasing attention for its potential to treat various gastrointestinal and even extragastrointestinal conditions in our beloved animal companions. Just like in human medicine, the trillions of microbes residing in the gastrointestinal tract of dogs, known as the gut microbiome, are thought to play a crucial role in influencing both the local and systemic immune systems by regulating inflammation and different populations of T cells, which are important immune cells. When the delicate balance of this microbial community is disrupted, a state often referred to as dysbiosis can occur. While there isn't one universal definition, dysbiosis generally implies a reduction in the variety of microbes present, a decrease in beneficial or commensal organisms, an increase in potentially harmful organisms, or a combination of these factors. This imbalance has been observed in veterinary medicine in connection with immune-related diseases such as chronic enteropathies, which are persistent inflammatory conditions of the intestines, and atopic dermatitis, a common allergic skin disease. Fecal microbiota transplantation is proposed as a method to restore what is termed normobiosis, essentially a healthy balance of gut bacteria. In human medicine, the Food and Drug Administration has already approved FMT products for the treatment of recurring Clostridioides difficile infections, a severe diarrheal illness. Furthermore, research suggests potential benefits of FMT in humans with inflammatory bowel disease, irritable bowel syndrome, and liver problems. In veterinary medicine, practitioners have been exploring the use of FMT for conditions like inflammatory bowel disease, acute diarrhea, chronic enteropathy, and parvoviral infections, a serious viral disease affecting young dogs. Given the well-established connection between the gut microbiome and the host's immune system, it's logical to consider that FMT could lead to changes in their intricate communication. Studies in humans and mice have indeed indicated that FMT can influence the peripheral immune system, sometimes leading to a reduction in pro-inflammatory substances in the blood and an increase in bacteria associated with anti-inflammatory effects and regulatory T cells. However, research in veterinary medicine has, until recently, not extensively investigated these potential changes in the peripheral immune system. One of the complexities in understanding the safety of FMT lies in differentiating between the clinical signs and immune changes related to an existing disease in sick animals and those that might be directly caused by the FMT procedure itself. Historically, studies on FMT in dogs have primarily involved animals already suffering from illness. Considering FMT as a therapeutic intervention, the researchers in the study we are discussing today recognized the importance of first understanding its effects on healthy individuals. This approach mirrors the initial phases of clinical trials for new drugs in human medicine, where safety and potential side effects are assessed in healthy volunteers. While investigations in humans have generally indicated that FMT is a safe procedure, with most reported adverse events being mild and self-limiting gastrointestinal symptoms, serious adverse events have been documented, albeit rarely. As the use of FMT expands in veterinary medicine, a comprehensive understanding of its safety profile in animals is crucial. Therefore, the study we are summarizing aimed to determine both the short-term safety and any changes in the peripheral immune system of healthy dogs following a single administration of FMT. To achieve this, the researchers conducted a prospective, non-randomized clinical trial involving 10 clinically healthy client-owned dogs as recipients of FMT and 2 clinically healthy client-owned dogs as donors of the fecal material. The recipient dogs received a single rectal enema of fecal material at a dose of either 2.5 grams per kilogram of body weight for the first dog and 5 grams per kilogram for the remaining 9 dogs. Over the subsequent 28 days, the dog owners recorded any adverse events they observed and monitored their dog's fecal consistency. On days 0, 1, 4, 10, and 28 after the FMT, the researchers collected fecal and blood samples from the recipient dogs. They also calculated a canine fecal dysbiosis index using a laboratory technique called quantitative polymerase chain reaction to assess the balance of the gut bacteria. The results of this study provided valuable insights into the short-term effects of FMT in healthy dogs. Notably, the researchers found no significant changes in several key variables. These included the complete blood count, which provides information about different types of blood cells, serum biochemistry, which assesses organ function, and the concentration of C-reactive protein, a marker of inflammation in the body. Furthermore, they observed no significant alterations in the levels of specific serum cytokines, which are signaling molecules of the immune system, including interleukins 2, 6, and 8, as well as tumor necrosis factor alpha. The study also found no significant changes in the numbers of different types of peripheral leukocytes, which are immune cells circulating in the blood, including B cells, T cells, cluster of differentiation 4 positive T cells, cluster of differentiation 8 positive T cells, and regulatory T cells. Finally, the canine dysbiosis index did not show any significant changes over the 28-day study period. While the study did not reveal significant changes in these objective measures, some mild and temporary adverse events were reported by the dog owners. These included mild vomiting in 3 dogs, diarrhea in 4 dogs, decreased activity in 2 dogs, and decreased appetite in 1 dog. Importantly, all of these mild symptoms resolved on their own without any medical intervention. In their discussion of these findings, the researchers emphasized that fecal microbiota transplantation, administered as a single rectal enema, did not significantly alter the evaluated variables in healthy dogs. They concluded that, in the short term, FMT was not associated with serious adverse events, changes in the peripheral immune markers they measured, or alterations in the canine dysbiosis index. The researchers compared their findings to studies in humans, where FMT is generally considered safe, with serious adverse events being rare. The mild adverse events observed in the canine recipients, such as diarrhea and vomiting, are also the most commonly reported side effects in human FMT recipients. The researchers suggested that the slightly higher percentage of dogs experiencing adverse events in their study compared to some previous veterinary studies might be due to the systematic use of a detailed scale to track these events, which may not have been as rigorously applied in earlier research. They also raised the possibility that the delivery method, a rectal enema, and the use of glycerol as a cryoprotectant in the preserved fecal material could have contributed to the transient diarrhea observed in some dogs. Glycerol, even when given orally, has been reported to cause diarrhea and vomiting in both humans and dogs. Interestingly, the study did not find significant changes in markers of inflammation like C-reactive protein or the measured cytokines. The researchers noted that the interpretation of cytokine levels in dogs can be challenging due to substantial variability influenced by factors like age, breed, and body condition. They also highlighted that their findings regarding C-reactive protein align with a study in dogs with inflammatory bowel disease treated with FMT, where no significant changes in this inflammatory marker were observed. Furthermore, the study found no significant alterations in the populations of different peripheral immune cells. This contrasts with a study in healthy human volunteers who received FMT capsules, where transient decreases in lymphocytes and certain types of T cells were noted. The fact that the canine dysbiosis index remained largely unchanged in the healthy recipient dogs suggests that the single FMT administration did not cause a significant shift in their overall gut microbial balance in the short term. 2 dogs did have a dysbiosis index above the normal range at the beginning of the study, and their scores fluctuated, but they did not develop any clinical signs of gastrointestinal disease during the study or in the long-term follow-up. The researchers acknowledged several limitations to their study. The number of dogs included was relatively small, which means the study might have been underpowered to detect subtle or less frequent effects. Additionally, the lack of a control group that received a placebo treatment makes it impossible to definitively attribute the mild adverse events solely to the FMT procedure. It's possible that some of these signs could have occurred by chance. However, the study was specifically designed to gather preliminary data on whether serious adverse events might be expected at the FMT dosages used, which are within the range commonly used in veterinary practice. The study also only investigated FMT administered via rectal enema, and the effects of FMT given through other routes, such as oral capsules, might differ. The researchers also pointed out that FMT might have localized immunological effects within the gut itself that were not detectable by measuring markers in the peripheral blood. Furthermore, healthy dogs might have a different response to FMT compared to dogs with pre-existing gastrointestinal disease. This study focused solely on healthy animals to establish a baseline understanding of safety. In conclusion, this study provides valuable data on the short-term safety of fecal microbiota transplantation via rectal enema in a small group of clinically healthy dogs. The researchers found that it was not associated with serious adverse events, significant changes in the measured peripheral immunologic variables, or alterations in the canine dysbiosis index. The mild and self-resolving clinical signs observed warrant further investigation, ideally in larger, placebo-controlled studies, to fully understand the potential effects of FMT in both healthy and diseased dogs. This research contributes to the growing body of knowledge surrounding FMT in veterinary medicine and provides a foundation for future studies exploring its therapeutic potential in a variety of canine conditions. Thank you for listening to the Veterinary Research Channel. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.